Podcast is now starting. All attendees are in listen-only mode.
Hello, everyone. My name is Sarah Kessel, and I'm an application scientist and lab manager here at the Nexalum Bioscience in the San Diego, California office. I'd like to thank you all for joining us today for our Celigo training webinar. The topic today is an adherent plate-based assay for cell cycle using the Viastain Celigo Cell Cycle PI reagent from Nexalum and the FCS Express software from de novo. And it will be presented by myself. All of our attendees will be muted throughout this duration of the presentation, but please use the GoToWebinar dashboard to type in any questions you may have as we go along. We'll have time at the conclusion of the presentation to answer any questions you may have. Also, the session will be recorded and you'll receive an email with a link to the recording so you can re-watch the presentation or pass it along to a colleague if you wish to do so. Without further ado, let's get started with the presentation. So typically this is an assay that is run on flow, where it may require a 6-well plate with a ton of cells per well, and you don't get a whole lot of replicates or dose response type curves. So I've been asked to create this in a plate-based assay for a 96-well plate. And so that's what you're going to witness today, so that you don't have to do any trypsinization and go into flow. It'll all be done in the plate with an imager. So just to give you an overview of what we're going to cover today, it'll be the plate setup and the staining. Then we'll cover how to acquire the images, analyze them, and export the data results for that image set. And then we'll import that into FCS Express without a template, meaning something that's already set up. We'll teach you how to make a template and then re-import that data using the template that we've just created. So this will be something that you can use over and over again for the next set of samples that you'll be running. So my tips for this assay type, I would recommend highly to have a positive and a negative control. For the positive control, I would have a drug treatment that is known to move the cells into a cycle for G1 or G2 phase. This will help you to be able to make sure that your data set is properly detecting a treated cell that has shifted or not. And then also for the negative control, you can use either media or a drug vehicle. So for the materials, you're going to need the Cell Cycle PI kit from Nexalum. You're going to want the ethanol to be ice cold. I just used my 80% ethanol and I put it in a bottle in the -20 freezer until I'm ready to use it. You'll need pipettes, and then today we're going to use a 96-well Greiner half-area plate with the catalog number 675090. You'll want to adjust your number and volume if you change to a different plate type. So for the assay setup, you're going to want to prepare your adherent cells in the 96-well plate. You'll also be doing all the drug treatment, staining, and imaging in this same plate. There is no trypsinization required for this assay. For the cell number per well, it's important to understand the proliferation rate for your cells and the length of your drug treatment so that at the end of your assay, you'll have 80% coverage of the well. So for example, today, I would want to have 8,000 to 10,000 cells at the time of fixation at the end of my experiment. If you have too many cells, they cluster close together and it makes the fluorescent reading difficult. And if you have too few cells, it just has less data points. So here's an example image of an ideal cell number density so you can see the proper spacing so the cells are properly identified and not having fluorescent overlap. So once you have your cells plated, then you want to adhere them overnight. The next day you can come in with your drug treatment that will induce the cell cycle arrest. And the example I'll show you at the end will have an 18-hour treatment. So the staining procedure is pretty simple. After your drug treatment, you're going to remove all the liquid, and then you're going to add ice-cold ethanol. For dumping out the liquid, I typically turn it over onto a trash can biohazard and then tap it onto a paper towel. Then you'll add your ice cold ethanol at 100 microliters, and then you're going to put it in the refrigerator for 30 minutes at the minimum, but you can go up to overnight. Remove all the liquid in the wells, and then add the stain for the cell cycle PI reagent, and then incubate in the 37 degrees incubator for the RNAs to digest the RNA. And then you're just going to image on Celigo. Very simple. Again, for me to dump out the liquid in the wells, I just invert it over a biohazard bag real quick, and then I tap it onto a paper towel to just dry it quickly. So now on the Celigo software in the Home tab, we'll click Create a New Scan. In the Setup tab, we'll click on the 96 well plate format, and we're going to choose the Griner plate. It is alphabetically listed on this list, and it's the 675090. It's a half area plate. And then we'll give it a plate ID. So here I have one already listed, so we'll make it called Cell Cycle PI Demo. And then you click Load Plate. The stage will open up and you'll put the plate on the stage, and then you click OK. The stage will move back into the system and move into the well A1 area. Now we're on the Scan tab, and today we're going to select the application in the Expression Analysis Suite, Target 1 plus 2s to collect the red and bright field channels. You can also do just one channel and do a red only, but today I'm going to show you how to acquire a red channel with a bright field channel so you can see your cells in bright field. But no analysis will be done on the bright field. So for target 1, we're going to select the red illumination. The exposure default starts at 10,000. That's always going to happen. So I know that this stain is going to need a little bit more exposure, so let's just bump it up to 50,000 and click snap so we don't have the illumination or the light on at all times. And you can see the cells are there, but they're a little out of focus and a little dim. So we'll just move over to a well that's C3 so we can register over on that well. And again, there's still a little out of focus. So let's click Focus Setup at the bottom. And this time we're going to check image-based autofocus instead of hardware because we want to have good, crisp images. And then we'll click Register Auto. So this means the system will actually study the cells in the image in fluorescence on every well until they're in focus and then take the picture. So here you can see that the images are moving up and down in Z. It will also do that same process when it's acquiring the images until it finds them in focus. And this is what they'll look like when they're in focus. So we'll scroll in, and what I want to do is now teach you how to set the appropriate illumination exposure. So we want to use this dialog down here. It says intensity to have the pixels on the object to be in the upper half of the camera range. So the camera range is from 0 to 255, and we want to be above 150. So let's just bump that exposure up to 80,000, and you can see we're just above the halfway mark for the majority of the cells. So that's just fine. And I might want to go just a little bit above that, but I typically won't go above 100,000 microseconds. If that's the case, then it just takes longer for the cells to, or the imaging to acquire on the whole plate. But for this reagent and this cell type, 90,000 looks pretty good. Again, you're going to use the intensity values down below to help you determine if you've got your cell object in the right pixel range. Okay, so we've set up the target 1 for red illumination and focus. Now we'll move over to target 2 and set up the illumination for bright field and change the focus because it'll be in a slight focus focal plane different than the red channel. So here they're a little bit blurry, so we'll go ahead and click Fine Focus. This fine focus is just going to move up and down in Z until it thinks that the cells are in focus or with really good contrast. And then the value that's right above where we just clicked says 20 microns above the red channel. So we'll click set offset, and that's going to tell the system when it acquires the bright field channel, it should move 20 microns above the Z plane where the red channel was acquired. And that's where this image will be in focus. And then you click apply. The apply will apply the illumination for every well. And then we select the wells that we want to image, and then we just click Start Scan. Now we move over to the Analyze tab, and while the first few image wells are being collected, I know there are some analysis parameters that automatically I used for this type of assay. One is the well mask, then separate touching objects, and then the pre-filtering, I keep the range for the red objects to be between 50 and 350. That means we'll exclude little tiny objects and really clumpy cells or oblong objects. And then finally, aspect ratio. Like I said, oblong objects will be removed if they're not a nuclear roundness. So those are the values that I have been finding to be working really well for this type of analysis on the stain. And then when we move over to target 2, because it's a bright field image, we really don't have any analysis on this page. We're going to turn the intensity threshold up to 255, which is the max. This is so we will not detect any objects in bright field. So let's take a peek at some of the images, or here's an image A2, and it will populate the screen with a bright field and a red fluorescent stain nuclei. And now let's scroll in so we can see how the graphic overlay for the red nuclei stain is looking. Okay, so it's looking pretty good. We've got the nuclei identified, but some of the nuclei are not being separated, even though I had checked off the separate touching objects. So let's go back to target 1, and you can see I have checked it. So I'm wondering, why is it not being applied? So let's go over to the bright field channel, where I know that we for sure should not be detecting anything. So let's turn on the target 2 and the graphic overlay. So since we are seeing the graphic overlay, that means it hasn't been applied, the settings that we typed in. So we just have to click either Analyze down below here or turn on the Auto Analyze. And that means anytime parameters will be updated on the fly. When you type it in, it applies it. If you don't hit the Auto Analyze or Analyze, it will not be applied. So that's why it was not being removed. Or here you can see on the target 1, the nuclei that are close together that were identified as 1 are now being separated into 2. So just remember, when you're going to make some analysis parameters adjustments, you either have to click Analyze or turn on the Auto Analyze checkbox. All right, so I'm happy with these settings. What I want to do now is save them so that if we come and do this assay again, we'll have an easy recall, because these are pretty much the standard settings that I've been using for this analysis, and I don't see much adjustment beyond that. So let's give it a name, the settings, and next time you run the plate, you can just automatically call these up. And then we'll click Selection and highlight the entire plate, because we want to apply all of these analysis settings to this entire plate. And then you can also click Analysis Settings and then highlight all of the wells. And clicking the space between A and 1 will highlight them all automatically. All right, so then we just hit Start Analysis, and the system will continue to acquire the rest of the plate's images and apply all the analysis settings that we just set up. So we'll just do a little fast forward for the image acquisition and analysis of this plate. You can see the counts now are coming from the nuclei of the red channel, and it's about 13,000. So even though I might have plated 10,000, the cells overnight probably proliferated just a little bit, so now we're above that. And now it'll give you a little bit of understanding cell density for this type of assay. Okay, so let's take a peek at this. So we have the cells in the whole well stitched together, And as you zoom in, you can see the bright field image behind the nuclei. How did the analysis settings turn out? So let's turn on the target one, and they look like they're properly identifying the nuclei. And then we have the bright field graphic overlay where nothing comes up because we chose to not pick up anything in the bright field image. Okay, that looks really good. Now let's move over to the edge of the well where we see the cells have clustered. This can happen a lot when you're plating cells and you have a either your incubator might not be level, or you pipetted in one direction and they ended up settling in one side versus the other. Or the plate may not be flat, and towards the edge of the wells they tend to have a different well level, so the cells might congregate on the edge. And on the other half of the well, you can see there's less cells. So this can happen, but rather than using the well mask on our Celigo software, I'll show you in FCS Express how we can use a well mask in that software, and it'll help you understand why over clustered cells can cause analysis problems in cell cycle. So this looks pretty good. I'm moving around and I'm pretty happy with this. So let's go back, and in the results tab, I want to export object level data. So that means an object piece of information, or every object's going to have a data exported. And we'll switch it over to the ICE with no image export format. And for report template, we're going to select none, but we're going to teach you how to make a report template. So then just click export. So the system will export the data into the folder that's selected above. And now we're going to open up the file in the de novo software FCS Express. And this is the 6 with the flow. And now first we'll create a new layout. In this white page, you'll see that it looks very similar to the Microsoft Office programs. So first we'll click on data in the ribbon, and then data list. And then we'll add a plus sign and grab the data that we just exported. So here is the export file. It's called Cell Cycle, which is our scan ID. And then at the very bottom of this folder, so those are all the well data, the very bottom is a file called object.ice. We'll want to select that one. And we'll move it over off to the side. And then we'll grab that file and drag it onto the white space. It'll ask us to select what kind of plot. It automatically defaults to heat map, so we'll keep that and click OK. So now it populates a plate layout for our wells. Next, what I want to do is create a density plot for the X and Y positions of each of the objects in the well. So this is where I'm going to teach you how to make a new different kind of well mask. So in the Insert tab, we're going to go over to the Density plot, and then you're just going to draw a diagonal right across the white space. Okay, it will populate every object that was detected in every well inside this plot. What we want to do is create an ellipse gate on top of this well, and we're basically going to only grab the objects inside this gate and exclude anything outside of it. So just dragging over the circle, and then it populates this text box. We'll type in WM for well mask. And then I changed the color to black because it's a little bit easier to see with all the other colors on this graph. Clicking OK. So now you can see we have a text box that says currently we got 62% of the objects detected. And you can change the size of the box by clicking the little squares and increasing the size or decreasing it. And then if you want, you can also move it around into the well to be better centered. It's your preference. But right now you can see that there are quite a few cells where it's red, that means there's a higher density. Where it's blue, there's less density. All right, now we're going to create a new plot by clicking on the Insert tab and then going to the Density plot. We're going to draw another diagonal over to the right side of that other plot. And this time, we're going to change the axis just by clicking on it from the Y area to the area, and then now to target 1 integrated intensity. All right, why are we doing this? This will help us to kind of clean up some of our data from when we create the cell cycle. We don't need every object that's kind of off into the higher integrated intensity. We need the majority. So now we're going to create a gate using the polygon icon, and then just click on the graph in the area that has the majority of the objects in this plot. Then I'm just clicking on the graph area, and then you complete it by clicking on the first one. The dialog box comes up, and then we're going to call it total. So this will become our denominator for each of the wells. And then I want the parent gate to be the well mass gate, and we'll change the color to black. Okay. So currently the gate is not gathering it from the well mask, so we're going to actually change it over here on the left to grab the data. So the plot going into this, the data going into this plot will come from the gate of the well mask. You saw that the density plot became a lot less objects. Okay. So now the information going into this plot is coming from the well mask gate on the previous plot. So now what I want to do is create a gate view box so you can see the hierarchy of the gates in the plots. Clicking on the Insert tab with the gate view and then drawing a diagonal across this space, you can see we have our 2 gates, the well mask and the total is within the well mask. Next, I want to create well gates grouping replicate wells together. So you don't have to have a plot per well, you can group them in replicates. So clicking on the gating tab and then highlighting or clicking on well, and then you just highlight over the wells that you want to group together. Then you give your gate a name. So this we'll call B456. And we'll change the parent gate to gather the data from the total. And the color doesn't really matter, so we'll just click OK. All 3 wells are highlighted. We'll just click off of that map, and then we'll do it again with the 3 wells below that grouping. Highlighting the wells, giving it a name. We'll call it C4, 5, and 6. You can name it whatever you want, but just for purposes of training, we're going to give it the name of the well numbers. And then don't forget the parent gate, and then OK, and then you just click off the map and it disappears. To have them show, you just right click on the map, and then in the dialog box it says show gates, select all. Now you can see the color-coded wells that we've just grouped in replicates. And in this gate view, you can see that those 2 new gates are now inside the total. Next, we want to create a multi-cycle DNA plot. So we'll go into the Insert tab and click on Multi-Cycle DNA and highlight a diagonal over the white space. And select in the dialog, we want Target 1 Integrated Intensity. This will be from our red channel. So a cell cycle plot histogram is displayed. And so now we want to actually have this plot reflect data only coming from the wells B4, 5, and 6. So we went into the gate tab and changed it so that the current gate in this cell cycle is looking at only those 3 wells. Now the histogram's kind of shifted to the left. If you want to change the axes, you can go click to the right click on your mouse, magnify resolution at the bottom of the menu, and then just highlight over the area that you want to have zoomed in on. So now the histogram is focused on just the plot area that has the data, but it's not looking too pretty. So this is where we want to use the graph right above us. here at the object X and Y positioning to see if we can improve it. So what I want to do is change the well mask gate so we can only collect data or show the data that's in the blue and green region. I want to exclude the area that has red because the cells probably are too clustered together and might be confusing the data displayed. So just by adjusting the well mask, you can see that all the graphs that are hierarchically below it are actually updating according to the new gate location. And you can see that our cell cycle area our histogram has improved significantly. So that's why we wanted to have a well mask created in the FCS software versus in the Celigo software. You just have a better idea. So let's change the title. So if you right click on the graph and then click Format, in this menu on the left, we'll click on Titles and then highlight the words that are in gray and then type in current gate or current well. We'll use current gate for now. And then in the Insert Token dialog drop down, we want to put in current gate. And then click OK. So now the title will be reflecting what is the gate that is displayed, or what is the data being displayed from what gate. So we have that. Now we want to right click again and we can show the statistics for the DNA cell cycle. And then I'm just going to move it below. And if you right click again on that table, you can turn off the different data displays for things that you may not be interested in. And you can basically have only the data that you want displayed. And then I just am going to make it look nice by increasing the size of the histogram. So if you want to now make another plot for the other set of replicates, you just highlight both those table and plot, and then Control C and Control V, copy and paste in another space, and then drag it over to an empty area. And now it's still on the gate for B4, 5, and 6, but you can change it by just clicking on the histogram and going into the Gate tab and changing the current gate to, say, the other replicate, which was C4, 5, and 6. And the data will display properly. The histogram and the table down below will update as well. So basically, you have created all the elements that you need for the cell cycle PI. And at this point, what I want to do is just kind of make it a nice report form, showing you how to center things. You just highlight and move it around. You can change the plot sizes. And then if you want to insert a title, you go into the Insert ribbon, text box, and then you can add a title. This is if you had more specific data that you wanted to put in there, an experiment title, you can do that if you want to keep it generic like we're doing here. And you can edit the text going into the text ribbon. So we're just making it look nice so you can have what the beginnings of a template would be for your future projects, future experiments on this assay. Okay. So now I want you to go into the Data tab and then click on Panel. Then we're going to click on the Add button. And then we're going to give the panel name a name. We'll give it a name called Celigo, doesn't matter. And then we'll increase the number of tubes to 1. And then we just click OK. And then we click OK again. This basically is telling the software when you import data in the future using this template, what order of operations of graphs do you need to apply. Now we're going to save this file as a template. So clicking Save, Save As. And then I'm just going to choose a folder location. I made this one up. It's FCS template. And then you want to give it a file name. I'm going to call it template because it basically doesn't have really an identifier that it is a template because it's just a .fa file when you see it, but we want to select the unlinked data, meaning it's just going to be a template without any data, and then click Save. But other than that .fa or unlinked data, you won't know that it's a template. So that's why I put template in the file name. So now what we can do is close the FCS program and then save that template in the Celigo database as a template for reporting this data. So here we're back at the Celigo software. We're going to go to the Home tab. And in the Home tab, we're going to go to the very bottom where it says Manage Data. And then we'll see at the upper portion of the screen, we have 5 tabs. We want to go to the 4th tab, what's called Report Templates. And in this middle portion of the screen, we want to click on the one that has a little blue and yellow icon, and we want to create a new one. So let's give this report template a name. Cell Cycle FCS Express Template Report. And then we're going to select that file that we just created in FCS. So let's go find it. We went out a couple of folders in Documents. FCS templates, there it is. We'll just select that and then click Open. Then we'll give it a plate category. This was a 96 well plate layout. The ICE no images is correct, and the application was target 1+2. So you know that it was 2 channels. So if you end up later on doing a 1-channel acquisition, you'll have to go through this whole process again, but with a 1-channel template. So in the description, I'm writing in there that it's a red and bright field image, so you know in image acquisition what was done. Here, once you click OK, the population list has now 3 files, and there in the middle is the one that we just added. Now what I want to go back to is the file that we just imaged and export with that template. So let's type in cell cycle so we can have all the scans that have cell cycle. Here's our item, the 5th one down. We're going to click on the 2nd scan, which was 2 channels, and then right-click on the analysis, and it says Review Result. That'll take us right back to the Results tab where we were a minute ago. Here we are with our data. And now what I want to show you is how to export into a template. So click on Export Object Level Data. We'll switch the format to the ICE no images. And we'll click on Report Template. There's our new template. And also auto open. So the file, once it's done exporting, will automatically open up FCS and populate the template with the data. It's a pretty big file, so it might take a minute. There is our report template and our data. All the data is plotted into the graphs automatically as we had designed a minute ago. So that's how you make a template, and that's how you export your data into a template. Next, what I want to show you guys is how to do add dose plot dose response curves to obtain your IC50s. Here I have an example plate layout where I plated some cells with 4 different drug compounds in triplicates for each of the drug doses. I have nicotazole, etoposide, carfentanil, what else? You know the other 2. And so we have our dose response curve down here. So you get the general layout of what we want to produce. So this has already been imaged, so we want to pull up that scan. And so here I have it called Cell Cycle PI for compounds, and actually I used the A549 cells. This was imaged with a single channel without bright field. And we just want to start with the exporting of the data into the FCS template. Now because this is a single channel, I used a different template that I had created earlier. So we'll grab the iSnow images, and this one would be cell cycle PI RNase 1, auto open, export. So when it opens, this is what it looks like, very similar to what we created, but I had added a couple more replicate and wells for the dose response. So if you guys are interested in having templates, I can either send you the ones that I've made or can help you make one. For the purposes of the graphs that we're going to make in the dose response, I'm going to move some of these graphs around. And what we want to put in the center portion are the cell cycles for each of these well replicates. So A is actually our 10 micromolar drug concentration. So that's a replicate of 3. And then B is the grouping in row B, and so on and so forth. I kept them all the same color because you'll see later when we do the 4 drugs, it'll make more visual sense. So the cell cycle graph here, we're just going to move it down to the bottom and then create replicates for each of the doses in the plate. So you remember before we just do Control-C, Control-V to copy and paste the replicate wells into the blank space. So we're just going to repeat this process so we have all 8 concentrations covered. So you can highlight all of them. And once you've created the plots, what you'll do is reassign the graph to the appropriate well sample. So this would be H, which is the bottom, and then you have G is the next one, and then we'll go up to F. So you just go up to the gate tab and tell it to collect the data from the F wells. And you just repeat that process for each of the well groupings. So we'll do that D, C. And as you're doing this, you can see the plot is updating, and you can see how the drug is actually adjusting the histogram profile for the cells, moving into more of the G2 phase as you get more concentrated drug. All right, so we have each of the concentrations represented with their cell cycle. We want to move the data into an Excel sheet. So in the Insert tab, find the new spreadsheet and then click on that. Give your spreadsheet a name. So we'll call this one Compound 2 because it's the second one in our plate. And then an sheet is displayed. We'll move it over to the right. And I just want to move it up a little bit higher on the page. Okay, so at this point, I want to adjust this sheet so it has our data in the right location. So in A1, we're going to type micromolar concentration. And then we'll right-click, or actually down below this, we're going to give our concentration of our compound. So this was 10 micromolar. And this is just like Excel sheet where you can add in formulas. So we'll do equals the 10 divided by 3 because I did a 3-fold dilution. And then you can grab the little blue dot and drag it down, and it'll keep the formula moving down. And you can right click and change the format of the cells by clicking Format Cells. We want to have a decimal point of 2. That's fine. So it's very similar to Excel, which is nice if you're familiar with Excel. And we'll change that 10 with a 2 decimal point as well, just to keep it consistent. And now on the graph, we're going to right click up at the top on the well, right click, and we will click Format this time. And I want the top row to be not a number. So we're clicking on spreadsheet. Use values in the first row as labels. So that first row is going to be a label in our graphs, in our graph. Now, what I'm going to show you now, I've now realized after making the video, is not the right thing to do. I'm actually highlighting all 4 graphs of A, B, C, and D, and then dragging it over into the Excel spreadsheet. Then we click on statistic token, and we're going to change the statistic to DNA cell cycle. And we're going to, while we're holding down the control button, click on %G, G2, and S. And then you click OK. Now the data is going to be displayed in columns according to those statistics, but later on you'll realize that the order that they're actually laid out in did not come in the A, B, C, and D order. It was actually the order of how the graphs were made, which was D, C, B, and A. So I'm repeating the process with G, H, E, F, G, and H. I'm going to move it down here at the bottom. It again, going through the statistics token, I want to display. On the statistics tab, you're going to click on histogram or cell cycle DNA. Then we'll hit the control button and then click on the G1, G2, and S percentages, and then click OK. So you get the idea. We're going to now delete that extra title and then highlight the wells and then drag them up into that location. So now we're going to create a scatter plot with this regression. So I'm going to click on the graph, click on scatter with regression, and then draw a diagonal across the white space. And now we have the beginnings of a plot, scatter plot. So we'll want to change both axes, the scale as well. So we're just going to right click, then click Format. And now we're going to click on Overlays. Currently in compound 2, we want to change the Y parameter to be G2. And you can hit preview to show the graph will update to the new display of G2 data. All right, then we're going to want to switch on to the axes, and we want to click on X-axis and change it from linear to log. And then click preview again. Then we'll click on regression fit. Currently it's linear, so we want to change that to the 5-parameter logistic. And then click preview. And then we can click OK. Okay, so we do have this graph, but the data is slightly backwards. And you'll see later I've corrected it. But just to note that you want to drag over each of the histograms onto the Excel sheet individually in the right order. So now I want to have the statistics. So right-clicking on the graph, you'll get the statistics. And here you have your min-max and the IC50 slope and the R-squared valued. Okay, and if you want to add some text box to, say you want to add a drug name or something, you can go into the insert text box and then type in a dialog. So this would be compound 1, and then you could type in the name of the drug if you wanted. Okay, And I want to update the title here because it's not just Celigo, it's going to be FCS Express as well. So if you're going to save this as another template, you remember the process that we went through to get to the template. You created a panel first, and then you saved it as an unlinked one. If you want to save it with the data, then you want to do it with linked. So now I want to repeat this process for all 4 of the compounds. In order for me to do that, I need to repeat the process and also create well gates. So rather than having you watch all of this, I'm going to actually show you the data already completed. And you can also name the pages for each of your compounds. So in this case, we'll call this one compound 2 because it's the second compound. So here is the completed version of the entire plate. Saw the exact same layout just a second ago, but now we have increased the number of well for each of the compounds and concentrations. You have your well gate gates down here, color coded, and each of the tabs here for each compound 1 through 4. And you can see how the wells are actually within the well mask first, and then it's going through the gate of the total gate here. And the data displayed on this page is coming for compound 1. And on this page, on this data display, I actually moved each of the wells over individually, or grouping wells over individually versus dragging and dropping like I did the 4 concentrations at once. So these are properly ordered. And you'll see that compound 2 looks much better than it did a second ago. And we have a good IC50 value there. Compound 3 and compound 4. That one really did move it into the G1 phase. That was the Dacto drug. So now you can see how you can create dose response curves for each of the compounds tested on a plate-based cell cycle PI stained assay. So if you had different cell types, you could just basically use the same template for each of the different cell type plates. So that brings us to the conclusion of our training session. I'd like to thank everyone for listening to our Celigo training webinar today. As I mentioned, we do have time for remaining in our session today to answer any questions you may have. This is your opportunity to ask a question about something that was presented today or any other question you might have, even if it's not related to the topic. All attendees are muted, so if you just type your question into the question bar on the GoToWebinar task screen, you can, we'll give everyone a little bit of time so you can type in your question. I'd also like to remind everyone that we'll be hosting another webinar, but on the celometer next month. So if you're interested on that, you can go to our website and register there. And also I encourage you to submit any topics for future webinars. If you're interested in anything specifically on this instrument or any other instruments that we have, any cell type or assays you'd like to see. And then also there will be a short survey that will pop up on your screen once we have concluded this webinar. And you can also type in topic suggestions there or email them to us at support@nexalum.com. So let me see if we have any questions. Okay, so far I'm not seeing any questions. Please type in any if you have any questions. Okay, I'm not seeing any questions coming through. So I guess then we'll conclude our session today. Again, I want to thank you attendees for taking the time out of your busy day to join us for our informational and training webinar. I hope it was beneficial for you. And as a reminder, you should receive a link to the recording to today's session via email so you can watch the presentation over again or pass it on to your colleagues. If you have any other questions you think of afterwards, you can reach out to us at support@nexalum.com. Thank you again, and I hope everyone has a wonderful day.